just trying to see where is the sharing thing in this.
One of the various approaches
Okay, fine, yeah.
to make vaccines against COVID-19. And as you know, last year our lives have changed because of SARS-CoV-2.
This is shared.
And what we are looking forward in 2021 is to come back to normalcy as early as possible. And for that, probably the only thing is preventive vaccine. Before we go to the vaccine, we all need to know how an immune response is generated to this virus. Because the vaccines utilize our own immune system to generate immune response. So SARS-CoV-2 is an RNA virus, and on its surface it has its major immunogenic protein, which is the spike protein. Once the virus enters our body, it gets into the antigen-presenting cells, where it is broken down into pieces, and the antigen is presented in contact with our MHC to the T cell. Once our helper T cells, what we call as CD4 T cells, see this antigen, they get activated and they provide help to B cells. The B cells are the ones which differentiate into plasma cells and produce antibodies. We are all aware that antibodies are protective to some extent, and the major mechanism by which antibodies cause protection is by neutralizing the virus or causing killing of the virus. In addition, the helper T cells also provide help to the CD8 cells. This is especially important in viral infections, where these cytotoxic T cells cause killing of the virus-infected cells. And as we have for other vaccines, we also want memory, that our body should know that we have been immunized against this virus, and if any one of us encounters the virus, we should have protection. And that is what we call as immunological memory, which should be for both B and T cells. So if you see here, the SARS-CoV-2 virus, initially it is, as is true for every viral infections or any infection, initially you get innate immune response, and then you get an adaptive immune response, and as I showed you, that you have T helper cells, you have cytotoxic T cells, and you have a special type of T cell which provide help to B cells, and you generate a memory cells. So this is showing you a speculative dynamics which have also been proven to a certain time point, that you have an antigen, the virus, which is shown in purple, and you generate specific T cells. And we hope that these T cells persist for years. And that has been shown for many other viral infections. Similarly, you develop antibodies, the antibodies wane off over time. And people have shown that in COVID-19 infection, there is waning of antibodies after 6 months. But you still have the memory B cells which persist for years. Vaccine development is a multi-step process, and it's not easy to develop vaccines. In the past, it used to take 10 to 20 years to get an effective vaccine. And the three most important steps are the preclinical testing of vaccines, where the scientists try to find out the best antigen possible, which will elicit a very robust immune response. And to further augment the response, we also use adjuvants, which activate the immune system and cause more activation and better immune response. So once we get the best antigen source and the best adjuvant, then they are tried in animal models. We generally start with small animal models like mouse, followed by ferret, which is a model for SARS-CoV-2, and then non-human primates. why it used to take decades to get a good vaccine. But in this era, with better knowledge of science and information from previous platforms which have been used for vaccine, we have been able to compress this period in a matter of less than a year. So this was an editorial in NHM in May 21, when it was thought that can we develop COVID-19 vaccine at the pandemic stage? The way the pandemic was spreading, would we be able to get vaccine at that speed? And then you have this project, light speed. And you've seen that in January 12th, 2020 was the day that the SARS-CoV-2 genome was sequenced. And in November 18, 2020, we had a vaccine which had 95% efficacy. I mean, this would have been unimaginable before 2020. And all this has happened because of support both from government, non-government organizations, collaboration between countries, and this partnership between researchers and the industries. Coming on the vaccine landscape, it just means that what are the various platforms or antigens which we can use for making vaccines. The nucleic acids, which carry all the message for all the proteins which the virus has. You can use DNA, you can use RNA. These are very easy to make because nowadays to synthesize nucleotides, sequence of nucleotides is very easy. But it has never been done before. Then you can have the light attenuated vaccines, which are the actual virus which have been made weakened so that it generates an immune response, but it does not cause the disease. Or you can make the virus inactivated by using some chemicals or other methods to kill the virus, but it still retains its property of having the antigens on its surface, but it cannot replicate. So inactivated viruses are very safe because they are already dead. Or you can use a small part of the protein, the subunit vaccines, like you're all aware of hepatitis B vaccine, which is a subunit protein vaccine. So these are very, very safe vaccines, but because you're using a small amount of protein, it may be that you don't generate a great immune response. And finally, you can fool the immune system by having an outer coat of a virus which is harmless. You can use measles, you can use rabies, and inside you can put the genetic material of SARS-CoV-2. That is viral vector-based vaccine. So, among these, most of these vaccines cannot be used as single-dose vaccines, while the replicating viral vectors or live attenuated vaccines probably can be used as single-dose vaccines. The DNA and RNA-based vaccines have never been used before, and so has been the non-replicating viral vector. And what we see today is the vaccines which are being used are the RNA-based vaccine and the non-replicating viral vector-based vaccine which we are all getting, that is Covishield. So these viral vector-based vaccines can be produced at a very high scale of production they can have. And similarly for the nucleic acid-based vaccines can be made at a very fast speed. So people have used all these advantages to make vaccines for COVID-19. This is the WHO website which tells us how many vaccines are in the pipeline. So you have, this is this week's data, that 173 vaccines are in preclinical development and 64 vaccines have moved to clinical development. That means they are in clinical trials. Of these, the majority are protein subunit vaccines. As I told you, they are most safe. And then you have non-replicating viral vectors, and then nucleic acid-based vaccines like DNA and RNA-based. We all want to have vaccine which is a single-dose vaccine, because you know the logistics of giving multiple doses to a large country and to vaccinate the whole world population is not easy. But there are only 20% of vaccines which are being tried as single-dose vaccines. Most of the vaccines which are in the pipeline this vaccine, the antibody is developed following the vaccine, you get memory CD4 cells, and finally, you also get that it does not infect the lung tissue. The virus is not able to infect. What I said that we also want a T cell response besides the antibodies. And in this is adenovirus-based vaccine. You can see that as the dose increases, low dose, mid dose, and high dose, there is good number of IF and gamma expressing T cells being generated, both at day 14 and day 28. And there is only a minor difference between low dose and mid dose. And the other thing which says how robust is the T cell response is how polyfunctional those T cells are. That is, how many different cytokines do these T cells produce. And if you see here that most of the T cells, CD8 cells and CD4 T cells which have been generated are predominantly polyfunctional, that they produce more than one cytokine. So that shows that we are generating a robust immune response by using this vaccine. And the government based on the clinical efficacy, but because the RNA vaccine has shown a great clinical efficacy in preventing infection. And this is our endpoint for vaccine. At one, we want to prevent infection. If we can't do that, can we prevent disease? If we can't prevent infection, can we prevent hospitalization? Can we prevent patients getting admitted to ICU? Or can we reduce mortality? And finally, for the society, can we prevent transmission of disease? We still don't have answers to all of them. The other issues which are there with vaccine are that we are unclear. How long is the duration of protection? Would it prevent an asymptomatic infection? Would it prevent transmission? And finally, would it cause vaccine-associated enhanced respiratory disease? This happens because when you develop, we want to develop only neutralizing antibodies, which will neutralize the virus. But if you also develop non-neutralizing antibodies, they can activate the macrophages and cause production of pro-inflammatory cytokines, which can cause tissue damage. Or these non-neutralizing antibodies can also cause increased uptake of the virus and increase in viral load sometimes. So what we have done so far, that we had a long way to go and we have reached quite far. And I think as a scientific community, we need to be proud of that. Till we get the final answer, this is our best vaccine. Thank you.
Thank you, Amita, for making it so simple for us to understand. Now, I would request Dr. Gagandeep Khan. He is one of the most eminent virologists in the country, who is currently in the Department of GI Sciences at CMC Vellore, and earlier she was the Director of the Translational Institute of the Department of Biotechnology. And of course, all of us now know that she is one of the few members of the Royal Society. Dr. Delaney, once again, thank you very much for joining us in this academic activity. Please start your presentation. She's going to talk to us about the early birds in the vaccines, which are already in use and in these 3 trials. Dr. Delaney, please.
Thank you, Dr. Kapoor, for the introduction, and thanks to everybody at SCTGI for having me here. I'm really going to touch on very up-to-the-minute news. There were some vaccines that made a lot of progress over the last couple of days. So what I'm going to talk to you about really builds on what Amita has just told us about where we are with the vaccines and the kinds of vaccines we have. So we heard about the mRNA vaccine, and there are 2 mRNA vaccines that are currently in wide use. The data from both of these vaccines became available in November of 2020. And one of the reasons that it became available so quickly was because they were funded very heavily by, in Moderna's case, by Operation Warp Speed, a challenge to deliver at scale, particularly in low and middle income countries. On the exploded map there, you can see which countries the Pfizer vaccine is licensed in. In green is countries where it has full licensure. In yellow is countries where it has emergency use authorization. It's also being given an emergency use listing by the WHO. So this is the vaccine with which we have the most experience, which is all of about 6 weeks since the rollout started. But millions of doses of this vaccine have been given. The Moderna vaccine, developed by a US company, is similar. It has 95% efficacy. Its storage requirements are less stringent, still requires -20 degrees. And this has also been approved by multiple regulatory agencies and given an emergency use listing by the WHO. Now, with Pfizer-BioNTech, if we look at the FDA's analysis, you can see here the data for 7 days after dose 2. So 5 weeks after you start the immunization program, you have about 95% efficacy. And as Amita showed in her slide, there's also efficacy, there's a good level of protection that occurs even after the first dose of the vaccine. Now, the other aspect of what we look at in addition to safety, in addition to immunogenicity and efficacy, is safety. And there are always safety concerns with new vaccines or with untested platforms. So in the trials, which included about 45,000 people and for whom about 38,000 people's safety data was evaluated, there were 4 cases of Bell's palsy in the vaccinated and 0 cases in the people who had received the placebo. Now, there were no cases of anaphylaxis that were seen in the trial, but subsequent to the vaccine being rolled out, at least initially, it looked like anaphylaxis was being seen at a rate of about 1 in 90,000 people initially, but it seems now that the rate is something like 1 in 150,000. And this is happening mostly in people who have a history of anaphylaxis before or have a history of very severe allergies. Just allergies to, occasional allergies to foods, to medicines, don't seem to be playing a role in predicting this. So this also speaks to the importance of having a really robust safety monitoring mechanism once vaccines are rolled out, because Bell's palsy looked like it might be a signal in the clinical trial. It doesn't so far seem to be one in the rollout. Anaphylaxis was not seen in the clinical trial, but is now being seen when the vaccines are being more widely used. Now for Moderna also, if we look at the protection, in all participants, it's about 94%. And if you look here at 65 and older, this is giving about 86% protection. Moderna's results are looking good. Moderna's vaccine is also being rolled out in Northern America and in Europe. Now, with Moderna, the safety signal seems to be somewhat less than is seen with the Pfizer vaccine. There is anaphylaxis that is seen with Moderna, but this seems to be in the order of 1 per 500,000 vaccinated individuals. So about 3 times lower risk than is seen with the Pfizer vaccine. Again, these are early days, and we will need to continue the safety monitoring to understand really how these vaccines perform. Now, if we look at vectored vaccines, how do vectored vaccines work? I think there's one thing that is important to spike primers to combine together to make the spike. On the surface of the cell that has been infected by the vector, you see both the entire spike protein as well as portions of the spike protein being displayed. When this happens, what you get is both an antibody response as well as a cellular response that results in the destruction of the cell that is viewed by the body as being an infected cell. Now, among the adenovirus vector-based vaccines, I'm sorry, my slide seems to have shifted a bit. The ones that are front and center are the Oxford AstraZeneca vaccine, also Covishield, which we are getting. And this is a vaccine that is given as 2 doses 4 weeks apart, and depending on how you look at which arm of the study, it has somewhere between 60 and 90% efficacy. It has received an EUA in the UK, restricted use approval in India, and yesterday was given full approval in the EU. There is a discussion ongoing about why Germany decided that the vaccine was not to be used in over 65-year-olds, but that is because the data from the study are not yet complete, and it's anticipated that when more data become available, there will be information to support that decision, support a revision of that decision. We also got data from the Janssen vaccine, and this is a recombinant adenovirus vector, which is at 26, and this is a vector that has been used previously for an Ebola vaccine, and this shows 66% efficacy. The Gamaleya Institute vaccine has been reported to have 92% efficacy, and this uses 2 different adenoviruses at 26 and at 5. CanSino uses an Ad5 vector, and this is a study for which data are not widely available in the public domain. Their phase 1 data was published in the Lancet and showed that if you had prior exposure to adenovirus, it resulted in a somewhat lower immune response. Now, I'd just like to walk us through some of the Covishield results. Shown in the map is where the serum Covishield AstraZeneca vaccines have been approved so far. And what I'd like us to focus on first is table 2C. This is the AstraZeneca efficacy, and this is for the standard dose and 2 doses of the standard dose. Now here you can see that if the vaccine is given at a dose interval of less than 6 weeks, efficacy is about 50%, and if the dose interval is greater than 12 weeks, efficacy is about 79%. Now, why does this happen? There's a lot of discussion about what is the immune response induced by the first dose and what is the immune response induced by the second dose, and why that dosing interval really matters. But if we look at the recommended schedule in India, that is 4 weeks apart. Now, the other thing that I would like you to look at is the comparison of Covishield and the Oxford vaccine made by AstraZeneca, and these are the data from the Indian participants, where you can see that Covishield is comparable to the results that are produced by the Oxford vaccine. You could argue that Covishield is actually a little bit better in terms of an immune response. However, since we don't have a correlate of protection, we don't actually know whether just the measurement of antibodies, whether they our IgG that is directed against the spike or neutralizing antibody really correlates with the clinical performance of the vaccine. It's hoped that we will be able to develop an immune correlative protection, but we don't have one at this time. Now, we got the results from the Janssen phase 3 study, and this was from almost 44,000 participants and over 400 cases. And what it showed was that the vaccine was 66% effective in preventing moderate to severe COVID-19 calculated to have 90% protection, 96% against the older coronavirus and 86% against the new variant, or what's called the UK variant. In South Africa, they had about 4,400 participants, and here the vaccine showed much less efficacy, about 50%, a higher efficacy in those who were not HIV exposed and lower in the ones who were HIV infected. And most of the infections here were from, well, about at least half, if not more, of the infections were because of the South African variant of concern, which is known as E1.351. So there's a lot that we've learned in the last month and a half about vaccines and their performance. There are many other vaccines